Hello and welcome to all my students. So today I am here to give you the ultimate recap of the chapter Molecular Basis of Inheritance. So far in this chapter, we have covered the major portion where I have made you understand what is the structure of DNA and what is the use of DNA. So coming to the first half of the chapter, that is dealing in with the structure of DNA beta, the most accepted structure of DNA was proposed by Watson and Crick in the year 1953. That is, DNA is not single-stranded, rather DNA is double-stranded and both the strands are helically coiled. Right? Now, further, if I talk about the use of DNA, that why DNA is the main genetic material, or who proved that yes, DNA is the main genetic material. So, regarding this, we have discussed so many experiments, and again today I'm going to give you a recap of all those experiments, followed by the use of DNA in making protein. So similarly, for this we have a central dogma which is going to explain you that how from DNA we can obtain protein, right? Hence it is done in 3 steps: replication, transcription and translation. So let's start with the ultimate recap. The topic which here we are going to cover in this lecture is the structure of DNA, who proved that DNA is the main genetic material, central dogma, and we are also going to cover the initial steps of central dogma, including replication, transcription and translation, right? Chalo. So let's start. So basically we are going to do a complete revision of this chapter. This is the main goal of our today's lesson. Now, first, starting with the structure of DNA, very, very important. So, beta, we have the most accepted model, that is the model proposed by Watson and Crick, right? So, there were two people, Watson and Crick, in the year 1953, proposed the double-stranded structure of DNA. Right? Now, Watson and Crick, for this, they also received the Nobel Prize. And let's see how the structure of DNA looks like. So basically, beta, Watson and Crick could propose the structure of DNA on the basis of the work done by other scientists. In short, you can say that whatever work was done by, or if these two people got the Nobel Prize, that is Watson and Crick, actually it was someone else credit who was received by them. So there were 3 different scientists, Maurice Wilkins, Rosalind Franklin and Erwin Chargaff, who actually gave some, we say most important discoveries, who did some most important discoveries in order to unwrap the structure of DNA. So let's see what these 3 people did, then what was their mistake, that why they could not propose the structure of DNA, and how these 2 people extracted the benefit out of it, right? So first of all, we are going to write the discovery of DNA. Now here basically, beta, in discovery of DNA, we are going to learn about 2 scientists, Maurice Wilkins, Rosalind Franklin and Erwin Chargaff. Right? Wilkins and Rosalind, beta, basically these 2 people, they proved with the help of X-ray crystallography that the chain of the DNA cannot be straight. Rather, it has to be helically coiled, and the pitch of the DNA, that means the distance between the 2 base pair on the DNA chain, has to be, yes, very good. The distance between the 2 DNA chain has to be 3 nanometers. So these 2 people told the DNA is helically coiled, and second, the pitch. That means the distance between two base pair is 3.4 angstrom, right? Whereas Shargaf explained the concept of complementary base pairing. Now, what is this complementary base pairing? Let's discuss. Complementary base pairing, right? Now, let's see beta. So, before we start with the work of these three people, little bit you should have the idea about the discovery of DNA, right? So, basically DNA was first discovered, this terminology came from the work of Friedrich Mischer, who was working on pus cells, right? Pus, you know, your WBCs. So basically this Friedrich Mischer scientist was working on the pus cells, where he found some negatively charged acidic substance which he called as nuclein. Later on, these nuclein were called as nucleic acid by scientist Altman. Typically when I talk about, now there are two nucleic acids, DNA and RNA. So when I talk about DNA, basically it stands for deoxyribonucleic acid. Definition wise, DNA is a polymer of nucleotide, right? Yes or no? Now, polymer means different units or same units, when they join together with the help of the bond, they form a long chain called as polymer. So, DNA is a polymer of nucleotide. In short, you can say that the basic unit of DNA is nothing but a nucleotide. Now, question arises in mind that, ma'am, what is a nucleotide, isn't it? So, I'll be first explaining all those points. Then we'll go back to the major discovery of DNA by Shargaf, Wilkins and Rosalind Franklin, followed by the work of Watson and Crick, right? So, over here, beta, when you see, so basically the definition portion, if I talk about of DNA, then DNA is a long polymer of, yes, it's a long polymer of nucleotide, or you can also say that basic unit of DNA is nucleotide. Now, question arises in mind, what is this nucleotide? So, basically, beta, each unit of nucleotide is made up of how many components? 3 components. So, basically, in a nucleotide, there are 3 components. Those 3 components are the sugar, which is the ribose sugar, a 5-carbon sugar. Then nitrogenous base. It is the variable part of the DNA because it can change. There are 4 nitrogenous bases which are possible: adenine, thymine, guanine and cytosine. So there are 4 nitrogenous bases which are possible: A for adenine, T for thymine, G for guanine, C for cytosine, followed by phosphate group, right? So these are the 3 major components of a nucleotide. Now question arises that, ma'am, how these 3 components are joined in order to make a nucleotide? So now let's see all the bonds involved in making a nucleotide, and then like that so many nucleotides are present which again joins with the help of some specific bond to complete the long polymer of DNA, right? Now if I have to draw a nucleotide, so the base is my sugar. Which sugar? 5-carbon ribose sugar. So see, here is my 5-carbon ribose sugar. First carbon, second, third, fourth, and to the 4th carbon is attached my 5th carbon, right? Now, this normally ribose sugar at 2nd carbon position, it is having H and OH. This is the normal arrangement of a 5-carbon ribose sugar. However, remember always, beta, whenever this ribose sugar is going to be utilized for making DNA, then the 2nd prime position will not have oxygen. So it will be called as deoxy. D means removal, oxy means oxygen, right? So instead it will just have hydrogen, no oxygen group. So this sugar is nothing but deoxyribose sugar. Clear? Yes. Now this is the first carbon, second, which is not having oxygen, third, fourth, and to the fourth carbon is attached the fifth carbon. Now, to this bit of first carbon, with the help of N-glycosidic bond, attaches the nitrogenous bases. Right? So what will you write now? So the next slide, you'll go and write. Now, sugar plus nitrogenous base. First of all, this association is known as nucleoside, and to make this nucleoside, the bond required is N-glycosidic bond, right? So which bond is required over here? N-glycosidic bond. Now, going back to the last page. Now see over here. Now, to the fifth carbon beta then attaches the third component, which is nothing but phosphate. And now this is the entire nucleotide, right? So now when you attach with this nucleoside phosphate group, then you actually make a nucleotide. And for the attachment of phosphate with nucleoside on the fifth carbon, the bond required is phosphoester bond, right? So one nucleotide consists of how many bonds? 2 bonds. Glycosidic, ester bond. Glycosidic bond is used to make a nucleoside, which is the attachment between the sugar and the base, and whereas phosphoester bond is required to attach phosphate group to the fifth carbon of the ribose sugar, hence making the complete nucleotide. I hope till here it's crystal clear to each and every one of you. Now like this, there are so many nucleotides present which again join to each other, forming the long chain of DNA. Now one more thing I told you beta, that in the structure of DNA, this nitrogenous base is variable. Why? Because DNA is made up of 4 nitrogenous bases which can be grouped into 2 categories, right? As purine and pyrimidine. Now, purines, there are 2, adenine and guanine, and pyrimidines in case of DNA are again 2, thymine and cytosine. These are 9-membered compound, whereas T and C pyrimidines are 6-membered heterocyclic compound. So if it is adenine in the nucleotide, it will be called as ATP. If it is guanine in the nucleotide, GTP. So CTP and TTP. So how many nitrogenous bases are possible? 4. So how many different nucleotides in a DNA are possible? 4. These 4 nucleotides only keeps on repeating themselves again and again for n number of times, making a long chain of DNA. Yes, got my point? So this is how you have to understand the basic unit of DNA, which is nucleotide. And DNA was discovered first by Friedrich Mischer in the pus cells. Later on, the name came from the Altman. Right? Chalo. Now coming back to the original discovery portion beta, as I told you that Maurice Wilkin and Rosalind Franklin, they were basically working on X-ray crystallography, right? And with the help of their X-ray crystallography, they found that DNA is not straight. Rather, it is helically coiled like this, plus the distance between two base pair, that means the distance between two nucleotide is 3.4 angstrom. Got my point? This was the major, milestone work done by these two people. Apart from this, when I talk about Erwin Chargaff, this person also did a great milestone work explaining that DNA is always going to show complementary base pairing. That means adenine. So according to Chargaff's rule, very, very important. This fellow observed that adenine, the number of adenine is always going to pair with thymine nucleotide. And guanines are going to pair with cytosine. In short, according to Erwin Chargaff beta, the amount of A plus G is always going to be equal to T plus C. Yes or no? If you just add these two equation number 1 and equation number 2, you want to get this thing, right? This is known as complementary base pairing. In short, you can write according to Chargaff rule beta, amount of purine is always equivalent to the amount of pyrimidine, right? So amount of purine is always equivalent to the amount of pyrimidine. So this is basically nothing but Chargaff's rule. Okay, thumbs up, understood. Now, using Chargaff's concept, Rosalind Franklin and Morris Wilkins' concept, Watson and Crick proposed the double-stranded structure of DNA. So next you are going to see Watson and Crick's model. So first point, they said the DNA is not single-stranded, rather it is double-stranded, helically coiled. So first point, that the strands of DNA are coiled in right-handed manner, right? The strands of DNA are helically coiled and double-stranded. First point. Second point, these two people said that both the strands are running anti-parallel. That means if the polarity of one strand is going to be 5 prime, 3 prime, then another strand polarity is going to be just reverse, which is 3 prime to 5 prime, right? So second point according to them is both the strands are anti-parallel, right? So over here, I hope you have understood this point. Okay, third, according to them beta, whenever the structure of DNA you will observe, nitrogenous bases will always project inside, and the backbone of this structure, the backbone of DNA is in the hands of phosphodiester bond. So third point you're going to write is nitrogenous bases. I'll show you with the help of the diagram also. Projects inward, right? So nitrogenous base projects inward and the backbone of the structure is nothing but phosphodiester bond. Right? Clear? Chalo. So now let's see that how this structure will look like. So there are my two strands of DNA. This is one strand whose polarity, if it is 5 prime to 3 prime, then the other strand will be helically coiled and whose polarity will be reverse, 3 prime above then 5 prime below, right? Now this is one turn. So according to Watson and Crick, one turn, the distance will be 34 angstrom and beta per turn, the total number of base pairs present will be 10. So how many base pairs will be present per turn? 10. That means now you remember what Wilkins and Franklin said, that the distance between two will be 3.4 angstrom. Automatically you can just calculate it, right? So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? So overall distance is 34 and the distance between two is 3.4 angstrom. So this was told by Wilkins and Franklin. Yes or no? Correct. Okay. Now next point, you can see that the nitrogenous bases are always projecting inward. I'm not drawing them like this, right? Rather I have projected them inward. So this was explained by them. Now next point which they also mentioned is that the backbone, these nitrogenous bases beta, they are joined with the help of phosphodiester bond. Plus one more thing, in complementary base pairing beta, adenine pairs with thymine with the help of two hydrogen bond. So if there's adenine, suppose if here it is adenine beta, then here will be thymine. If it is guanine, cytosine. And how they're attached to each other? With the help of hydrogen bonds, right? Adenine pairs with thymine with the help of two hydrogen bond, whereas guanine pairs with cytosine with the help of three hydrogen bond. This is the major information which you have to keep in mind, that here there are two hydrogen bond and here there are three hydrogen bond present, right? Chalo, very good. Now this is about the Watson and Crick structure. Now apart from this, these people, they proposed one more concept called central dogma. Very, very important, because the first major portion of the chapter is understanding the structure of DNA. If you have really understood it, then the second is the function of DNA. कि भाई इसका क्या काम है? Why it is so important? And that importance of DNA will be very clearly understood with the help of central dogma proposed by Watson and Crick. According to them, beta, the main job of DNA in any body is to obtain proteins, correct? But remember, this cannot be done directly. Rather, it depends upon three peculiar steps. The first step is known as replication, where from DNA you obtain DNA. Copying of DNA is known as replication, right? Then from DNA we try to obtain another nucleic acid by the name RNA. This is known as conversion of DNA into RNA, is known as transcription. So this is the second step. First step, second step, and then from RNA basically beta we obtain proteins, and the third and the last step is known as translation. So first, second and third, right? Now before we start with the central dogma concept, we have to do some basic experiments which explains that why out of two nucleic acids or three chemicals, that is DNA, RNA and protein, why only DNA is considered as the main genetic material. So the unequivocal proof, the best experiment to prove that yes, DNA is the only genetic material was given by Hershey and Chase, right? So the next you are going to write over here is experiment to prove that DNA is the main genetic material, right? And in this, the unequivocal proof means 100% guaranteed proof was given by two scientists, that is Hershey and Chase, right? So these were the two scientists who basically they were working on two, or they took two organisms to prove this. One was they took a bacteria called E. coli, plus they took a virus, bacteriophage, right? And in 3 steps they proved that yes, DNA is the main genetic material. Now let's see what was their experiment. So basically, beta, they took two viruses. First virus whose capsid, that means the capsid, the body of the virus, which is made up of protein, was replaced by radioactive sulfur S35, and in second virus beta, the genetic material was replaced by P32. So both are the radioactive element. Now you are having two viruses, A virus whose body is having radioactivity and B virus whose body is free from radioactivity, but yes, the genetic material is having radioactivity, that is phosphorus. Now, whatever will enter inside the one, where there's one very huge concept that whenever bacteriophage virus attacks a bacteria, its genetic material only goes inside. Otherwise, the remaining portion apart from the genetic material stays outside. One thing. Now, suppose if protein is the genetic material, agar protein genetic material hoga, then protein, that means sulfur S35 will go inside. However, if DNA is the genetic material, then P32 will enter inside the bacteria. And this is the only thing which we have to detect over here. We just have to detect that what is entering inside the bacteria in the form of genetic material, whether it is radioactive sulfur or whether it is radioactive phosphorus. So we just have to make out this thing. And for this, they are having three identical steps. Oh, sorry, 3 steps. The first step is known as infection. Infection means attaching the virus with the bacteria. पहले infect कराओगे, yes or no? Suppose these are my bacteria. This is A bacteria and this is B bacteria, right? Then you will make both your viruses attack on them. In first, S35 is there, and in second, the genetic material is having P32. This is first step, infection. As soon as the infection will be done, the genetic material of the virus will enter inside the bacteria. Right? Now here I am showing you that genetic material is DNA. That is why phosphorus is going inside. But when Hershey and Chase were doing this experiment, they were not aware about it. So they were just doing the experiment. After infection, the second step is blending. Blending means what? Now the infection is done. Now whatever we wanted to enter inside the bacteria, it has entered. That is the genetic material of the virus. Now we want the separation. कि भाई अब जो भी रह गया है, whatever remains of the virus are present on the body of the bacteria, it should get detached. So that is known as blending. So blending means what? Basically, beta, blending means removal of the viral remains from the bacteria. Correct? So now you're just going to remove all the viral remains from the bacteria, and the last step is to detect that whether the remains are having radioactivity or the bacteria is having the radioactivity. Correct? So the third step to prove this is centrifugation, that is density basis separation. Right? So in centrifugation step, we will come to know that phosphorus, which was there in the genetic material DNA, has entered inside the bacteria, whereas sulfur, radioactive sulfur, which was present in the protein, that is body of the virus, is in the remains. It has not entered inside the bacteria, proving that not proteins, rather DNA is the main genetic material. Right? So centrifugation जब हम कराएंगे, that means in A case, radioactivity is basically beta detected outside the bacteria, whereas in B case radioactivity is detected inside the cell. Yes or no? So have you all understood this simple concept, A case and B case? Okay, so if you have understood, so this is about the experiment which proves or which gives us 100% confirmation that yes, DNA is the genetic material and not your RNA or protein can be a genetic material. Right? Now, when we came to know, a final conclusion ho gaya hai, we all have understood that okay, DNA is the only main genetic material. So question arises that how this DNA is then converted into protein. And the first step towards central dogma is replication, right? Okay, yeah. Before we start with replication, one more simple concept I want to explain, that is packaging of DNA. Now, you know, beta, that DNA is so long. In human, if I talk about, it is approximately 2.2 meter long. So, DNA and your cell is so small. It's, hardly some 10 to the power minus 10 meter, and your DNA is 2.2 meter long. So, how this long DNA can very easily reside inside the nucleus of such a small microscopic cell. So that topic we study under packaging. I'm not going to tell you much about it, just little bit introduction, that's it, right? So now here I'm going to talk about, before I start with central dogma, let's do a little bit about the packaging of DNA, right? So when I talk about packaging of DNA, especially in eukaryote, so why it is necessary, why this packaging is necessary? Because DNA is too long and the cell inside which it has to be kept, it's very, very small, it's microscopic. So how such a long DNA will fit inside that such a microscopic cell? Of course, when you package them, like you have wool now, like wool, if it is open, it's difficult to keep inside the cupboard. Otherwise, it will go here and there, all the threads. But what your mother does, she simply makes a ball out of it, she packages it, and then she throws inside the cupboard, and then it becomes so small that everywhere or anywhere it is kept inside the cupboard, no issues. Similarly, over here also, right now our DNA is present like an open thread, and we can't keep DNA like an open thread. So what we will do, we'll tie this, and this tying is known as packaging. So for packaging of eukaryotic DNA requires the set of basic proteins called histone proteins. Very, very important, right? So basically, beta, packaging of DNA in eukaryote involves histone proteins. These are basic proteins, positively charged proteins, right? So basically these are positively charged proteins and made up of two amino acids, lysine and arginine, right? So this packaging of DNA in eukaryote involves histone proteins, which are positively charged proteins made up of two amino acids, lysine. Sorry. Yes, lysine and arginine, correct? So these histone proteins, they basically make a ball-like structure called nucleosome, and the number of nucleosomes they attach like a string called chromatin fiber. Chalo. Okay, now coming to the major portion of central dogma, and the first step of central dogma explains you रेप्लीकेशन. ठीक है भाई. अब क्या होता है रेप्लीकेशन? रेप्लीकेशन मीन्स कॉपीइंग ऑफ डीएनए, मेकिंग डीएनए फ्रॉम डीएनए. सो रेप्लीकेशन इज नथिंग बट copying DNA or making DNA from DNA, and this is done with the help of most important enzyme called DNA dependent DNA polymerase. क्यों भाई DNA dependent? Because from DNA you are making DNA, and polymerization means formation of polymer. So DNA is nothing but a polymer. So replication is nothing but simply copying of DNA from DNA. And for this, the most important enzyme is DNA dependent DNA polymerase. First thing. Second thing, beta, how does it happens? So it happens semi-conservatively. So there were two people called Michelson and Stahl, right? These two people basically beta proved that DNA replicates semi-conservatively. Now I'll tell you what this means, but first write it. According to them, the DNA replicates semi-conservatively. Now semi-conservative का क्या? Semi मतलब 50%. Conservation means as it is, keeping it as it is. Conservation, right? Protection. So suppose this is my DNA. These are the two strands of DNA, both running anti-parallel to each other. Now, here if I want to say that DNA replicates semi-conservatively, so what I mean to say is, first of all, during replication, beta, both the strands of the DNA will participate, both will act as a template, and from this one DNA chain, in future, you are actually going to obtain two daughter DNA. So one, you are going to obtain two, and out of these two daughter DNA, if you see, one strand is completely same as that of parent, and second is the newly synthesized strand. Can you all see this thing? That in case of semi-conservative replication of DNA beta, if this is my parent DNA, right? If this parent DNA undergoes replication and produces new daughter DNA, so from one parent we actually obtain two daughter DNA. ठीक, पहली बात. दूसरी बात, if you have seen that both the daughter DNA are having one chain same as that of parent, that is yellow color, and second chain is completely new. So that means you have conserved the parent DNA by 50%. आधा पुराना दिया है, आधा नया दिया है. So 50% of the DNA is new in case of daughter and 50% is old. This is known as semi-conservative replication. That means over here, daughters are having 50% old strand as parent strand and 50% new strand. Right? So this is the actually semi-conservative replication, and now be it prokaryote or eukaryote, everywhere beta, DNA is going to replicate in the same manner, and it was very experimentally clearly proved by Michelson and Stahl. Now, how does it happen? How this semi-conservative replication occurs? So for this, we have a whole mechanism, which over here, I'm just going to explain in brief. So the first step of replication is looking for the ori. Ori is known as origin of replication. I'll just explain with the help of the diagram, right? Suppose this is my DNA. I'm showing the prokaryotic replication. So in case of prokaryotes, the DNA is double-stranded and circular. And now every DNA has their ori, that is the origin point of replication. Now this is the point from where replication has to start. It is this ori is recognized by an enzyme called helicase, right? Now this helicase enzyme beta attacks the ori, it unwinds the DNA chain, and as a result forming a Y-shaped replicating fork, exposing all the nucleotides, right? And then primers are added on the initial side, to which DNA polymerase 3 comes and continues the replication following the complementary base pairing rule. So in short, just remember beta that replication, be it in both prokaryote or eukaryote, it is simply it is simply done by semi-conservative method. First point, the most important enzyme required for this process is DNA dependent DNA polymerase. Third, in case of prokaryote beta, this whole mechanism process is actually taking place in the cytoplasm of the cell, whereas in eukaryotes, it is taking place in the nucleus of the cell, right? Next point, then for beginning of the replication, there are some specific sequences present called ori, from where helicase enzyme unwinds the DNA, opens it to form a Y-shaped replicating fork, exposing the parental strands, to which DNA polymerase comes, reads and synthesizes the new complementary strand, completing the process of replication, right? Now, once the DNA is replicated, then we move towards the next step of central dogma, which is nothing but transcription, right? Now see beta, basically what happens in case of transcription? From DNA, you are obtaining RNA, and for this job, the enzyme required is RNA polymerase. There in replication, the enzyme was DNA polymerase, but here the enzyme is RNA polymerase. Why? Because in replication, you were synthesizing DNA from DNA, but here you are synthesizing RNA. So what polymerization has to be done? Polymerization of RNA has to be done. Therefore, the enzyme is RNA polymerase, right? It is also taking place in the cytoplasm of the cell for prokaryote, but in eukaryote, it is taking place in their nucleus. So this is the second important information which you have to remember. Moving towards the third beta. Now, unlike in replication, you have seen both the parent strands are participating, acting as template, but here out of the two DNA chain, right? Only one of the chain whose polarity is 3' to 5' will act as template. That means out of the 2 strands of DNA, only one participate in transcription, whereas in replication, both the strands were participating. So this is one more information which you have to keep in mind. Now basically beta, RNA are of 3 types: mRNA, tRNA and rRNA. All these 3 types of RNA are actually synthesized by transcription, and all the 3 types of RNA are highly involved in translation process also. So next you're going to write is, there are how many types of RNA involved? There are basically 3 types of RNA involved. That is mRNA, tRNA and rRNA, right? M stands for messenger RNA, which carries the information that, okay, which type of DNA has to be synthesized when, which type of protein sets has to be synthesized when. tRNA, which is also known as soluble or adapter RNA. It is also having the same job. It brings the right amino acid to the right codon on the mRNA for the translation purpose, and rRNA helps in synthesizing the ribosomes. In short, all the 3 RNA are then involved in the next step of central dogma, which is translation. So what is translation? It is the mechanism of protein synthesis from RNA. And where does it takes place? Be it prokaryote or eukaryote, in both of the cells, this translation process is taking place in the cytoplasm itself. So in the cytoplasm, right? Clear? So wherever the process is taking place, it is just taking place in the cytoplasm. Now, before this, there is one more concept which is very important for you all to know. That is genetic code, right? So the genetic code concept was very well explained by scientist George Gamow. Now, इन्होंने क्या किया? He gave us an idea that how you have to read the mRNA chain. So mRNA chain is made up of so many nucleotides. Now, how this mRNA is carrying the message? What is that message? That all concept was given by this fellow. इन्होंने हमें क्या बताया? This fellow told that genetic code is triplet. Triplet why? According to him, if an mRNA chain is made up of n number of nucleotides, right? Okay, remember beta, RNA is always single stranded, like DNA is double stranded. So RNAs are basically all RNA, be it mRNA, tRNA or rRNA, they are always single stranded, right? Yes, so they are single stranded. Now, according to George Gamow, the 3 nucleotide in a continuous manner makes one codon. This is another codon, this is second codon, third, fourth codon, right? And they're non-overlapping. If one nucleotide you have utilized in making one codon, the same cannot be utilized for making the second codon, right? So these are making codons, and according to them, one codon is coding for one amino acid. Clear? So this concept of genetic code being triplet, question can come You have to remember it was given by George Gamow, and later on experimentally it was proved by Matthias Nirenberg and yes, Matthias and Nirenberg. These two were the ones who actually proved that genetic code is yes triplet. However, its first concept came from the work of George Gamow. Triplet means 3 sets of nucleotide in a row on the mRNA chain makes one codon, and then that one codon is responsible for coding one amino acid. Clear? And then came your translation, where you have seen that protein synthesis takes place, and ultimately those proteins are responsible for controlling your phenotypic expression. तो फिर इससे हमें क्या समझ में आता है? We definitely came to know that if DNA is a main genetic material, if I am having DNA, then the DNA is doing nothing. It is simply converting itself first into protein by how many steps? 3 steps. Replication, transcription, translation. And with all these 3 steps, once the set of proteins are involved, then those proteins are then utilized to to do so many metabolic activities, and then controlling all our body expressions, right? So this chapter is molecular basis of inheritance, where you study cell at molecular level. What that means, what the cell is made up of, and one of the major component of cell is the nucleic acid, that is DNA and RNA. Both can be genetic material, but apart from these two, which becomes the genetic material? DNA. But DNA, can it work alone? No. It is highly dependent on RNA to obtain protein, and this is very well explained by the central dogma. Clear? Understood? Chalo. So yes, that's it for this revision of, or the ultimate recap of molecular basis of inheritance chapter. I hope that you all have found it helpful. So if you really want to enjoy such lectures in detailed manner, then yes, come join the batch, and there you will surely get the detailed description. So this was the ultimate recap of the chapter. Thank you for listening to me. Keep watching more and more amazing ultimate recaps in the upcoming time.